So our next presenting company, this is a perfect segue. Aastrom, as you heard this morning, is developing autologous stem cell therapy, ixmyelocel-T. Here to present the story is President and CEO Nick Colangelo.
Colangelo, close.
Colangelo, sorry. Thanks.
Thanks. It's a pleasure to be here. As you mentioned, we'll skip the safe harbor, but Aastrom is or has developed a leading autologous cell therapy platform that generates patient-specific multicellular therapies utilizing our highly automated and highly scalable GMP system. Our initial product or lead product is ixmyelocel-T, and that's a highly differentiated product whose primary effector cells play a key role in tissue repair and regeneration. We're focused on large unmet medical needs, particularly severe ischemic cardiovascular diseases. We've been in 400 patients to date, and the product has demonstrated that it's well tolerated in patients, and we've seen consistent positive efficacy signals. We believe our lead indication, which is the treatment of advanced heart failure due to ischemic DCM or dilated cardiomyopathy, which we talked a lot about this morning, presents a substantial commercial opportunity, and I'll explain that in a moment. And we also have an orphan drug designation for that indication. So as Rhonda explained this morning, ixmyelocel-T is a highly differentiated cell therapy. And the reason that's the case is that it's basically comprised of two key cell populations, mesenchymal stromal cells and M2-like macrophages, that are known to play key roles in tissue repair and regeneration, both individually and collectively. In fact, there are publications coming out now that mesenchymal stromal cells actually mediate their biological activity through macrophages through the upregulation of IL-10. So that's a very important synergistic component of this product. And basically, we're taking bone marrow from patients, expanding those two key cell populations to form ixmyelocel-T. So as has been mentioned throughout the day, again, these cell populations have their therapeutic effects both through paracrine effects or trophic effects of their secreted factor profile as well as their direct cell activities. So these populations have upregulated IL-10. They produce massive amounts of IL-10, IL-1 receptor antagonists that have potent anti-inflammatory effects. They also have secrete factors that are involved in tissue remodeling, like MMPs and TIMPs, as well as potent angiogenic factors such as VEGF, FGF, and so on. So it's a very effective cytokine and growth factor secretion profile. They also have cell-dependent activities that Rhonda talked about, so direct activities of phagocytosis, clearing away cellular debris that causes inflammation, efferocytosis, where they get rid of apoptotic cells, and then remodeling of extracellular matrix. So these combined activities, the trophic effects and the direct cell effects, lead to the therapeutic effects of tissue remodeling, promotion of angiogenesis, and reduction of inflammation. So our therapeutic platform provides for a very efficient cell collection, production, and delivery of the product. As I mentioned, we basically do a 15-minute outpatient bone marrow extraction procedure. The bone marrow is sent to Aastrom's facilities in Ann Arbor, where we manufacture the product in single-use disposable bioreactor cassettes that are part of a fully closed automated GMP manufacturing system, which is highly scalable. It's a modular system that's highly scalable to meet future production demand. So when we talked about industrializing the production or manufacturing process this morning, this system is clearly industrialized and can be expanded really virtually without limit. Basically, the finished product, the final product is released according to finished product specifications that have been approved by the FDA as part of our clinical programs. We basically FedEx overnight to the physician the finished product in a qualified temperature-controlled container that's ready to use when they receive it. So there's no freeze-thawing, there's no refrigeration, there's no reconstitution. There's a 72-hour shelf life, and it basically can be administered via syringe or standard catheter system. So it's a very streamlined logistical process for an autologous therapy program. So this, based on the attributes that I've described already, this platform offers the potential for multiple therapeutic applications. Anywhere where you have tissue damage, it's a relevant product. We're clearly focused, as I mentioned earlier, on cardiovascular applications, but it, again, has broad applications in fibrovascular diseases, et cetera. Our main focus right now is developing ixmyelocel-T to treat advanced heart failure due to ischemic dilated cardiomyopathy. So, as I'll talk about in a moment, we've initiated a phase 2b placebo-controlled study in this indication. So there's been a lot of talk about the heart failure market today, and luckily my figures match up with Dr. Miller, so that's a good thing. But basically, as he described, heart failure represents a significant unmet need and a growing public health problem. The estimates are anywhere from 5 to 7 million patients currently suffering from heart failure, and this is projected to grow substantially. The costs currently are in the $25 billion range and expected to triple to nearly $80 billion by 2030. So it's a serious issue and one that obviously has a lot of people's attention. When you look at ischemic DCM, it's basically the third leading cause of heart failure and the leading cause of heart transplantation. So the disease is characterized by the weakening of the muscle, enlargement of the heart chambers, and basically an inability to sufficiently pump blood. And, 60% of DCM is of ischemic origin, and these patients, as we talked about earlier, really have no other treatment options. They're on a path to an LVAD or a heart transplant, and that's it. So there's clearly an unmet medical need here. And if you look at the disease progression, a majority of the advanced heart failure patients that are refractory to medical therapy, as we talked about earlier this morning, have DCM. And again, 60% of that is of ischemic origin. These patients basically have maxed out on prescription drug, ACE inhibitors, hypertension medicines, et cetera, as well as medical devices. They're typically no longer candidates for revascularization procedures like bypass or angioplasty. So these guys are headed, or these patients are headed towards LVADs and heart transplants. So we think this offers a significant commercial opportunity as well. The number of ischemic DCM patients is growing. We believe we have a first mover advantage. We have an orphan indication in this, and the pharmacoeconomics clearly work in our favor. LVAD procedures are a couple $100,000. Heart transplants, estimated costs are in the $1 million range. So we think we'll be able to obtain premium pricing and have an opportunity to derive substantial revenues in this indication. So we also think there's a strong scientific basis for moving forward in this indication, both based on our preclinical data generated to date as well as our clinical data, which I'll describe in a moment. So this is just a representative example of our preclinical data. It's, as you can see, ixmyelocel-T significantly and reproducibly reduced tissue damage in a murine non-acute infarct model of heart failure. So statistically significant results in terms of the efficacy outcome, which is reducing infarct length, and also in the histology panels on the left, you can see that compared to vehicle and control groups, that ixmyelocel-T not only reduced the damaged tissue, but you'd think it actually helped with tissue repair because that's a pretty healthy-looking left ventricle, which is the circle in the middle. From a clinical perspective, we've conducted 2 12-month open-label phase 2A studies in DCM. And the objective of those studies was really to help guide the further study of this drug in this patient population. So we had 3 objectives. Obviously, you want to demonstrate safety of ixmyelocel-T in this patient population. We wanted to look at alternative delivery approaches and select the appropriate patient population. So we had a surgical study in ischemic and non-ischemic DCM patients, as well as a catheter study in those same patients. And from a safety perspective, as you'd expect with an autologous therapy, the product is very well tolerated in this patient population. Post-surgery and when delivered by catheter, the incidence of AEs is similar between drug-treated and placebo groups. And we saw that catheter administration improved patient tolerance. From an efficacy perspective, we were looking at real endpoints, so 12-month MACE events in these studies, and you saw that in the ischemic population, there was a significant reduction in the percentage of patients that had MACE events at 12 months compared to the non-ischemic patients. And we saw consistent positive trends. Again, these were small studies of about 20 to 30 patients, but we saw consistent positive trends in several other efficacy parameters related to symptoms and function, including increases in ejection fraction, increase in 6-minute walk test performance, and improvements in New York Heart Association class. So we think there was a strong preclinical and clinical basis for moving forward in our phase 2b study. And clearly, the results in the ischemic population with catheter administration formed the foundation for moving into our phase 2b study. So this is a multi-center, randomized, placebo-controlled, double-blind study in 108 patients at 30 sites in the U.S. and Canada. And again, the drug will be administered via NOGA MyoStar catheter. The key endpoints are registration quality endpoints, where we're looking at MACE events at 12 months. We'll also look at secondary efficacy parameters related to clinical, functional, structural, and symptomatic outcomes at 3, 6, and 12 months as well. So that we enrolled our first patients in that study earlier this month, and I'm happy to report that we treated our first patient today, and we'll be treating more than one this week. So the study's off to a good start. We expect to have it enrolled within a year. Again, it's a 12-month endpoint, so we expect to have data in the second quarter of 2015. And so, in closing, and I think we have time for a couple questions, if you think about what we discussed this morning as the critical success factors for cell therapy, it was, scalable and cost-efficient manufacturing, a real commercial opportunity, and, real clinical results. And I think we meet all three of those. In particular, with the DCM population, this is a well-defined population at a well-defined point in disease progression, and that's why we're excited about the potential for this product in that opportunity.
Great, Nick. Come on over and sit down with me. I appreciate you going into all the detail on DCM because I think that's an indication people may not be as familiar with. And I don't want to beat a dead horse here, but there may be some people here who don't fully understand all the factors that went into the decision to halt the CLI study. So while we're here, while we're public, I want to give you a chance to kind of walk through that. Was this really just a factor that the enrollment criteria were too stringent and you just weren't finding enough patients? Did the cost of the trial factor in? Tell us a little bit more detail, because I know that was a very difficult decision that you faced.
Well, yes, it was a very difficult decision for a number of reasons. I mean, these are patients who, as you heard earlier, have, severe conditions that result in amputations and so on. So, from a patient perspective, we didn't take the decision lightly to walk away from the study. But, I think I can contrast it to the DCM opportunity where, it wasn't necessarily a cost factor, the cost of the study. It was just the length of time to enroll a 600-patient study in a pretty tough disease, and is that really something we should be doing as a small market cap company? It was hard for me personally coming in within the first month there and making that decision along with the team, but we had been looking at options for quite some time. So it was really solely a business decision. We had great phase 2b data that would support moving forward, and our hope is that someday we'll be in a position to do it. But from a business perspective, if you think about what's the most efficient and cost-effective way to prove the principle and potential for this product, it wasn't the CLI study. It's a DCM study, and that's why we decided to move forward and invest our resources there.
Great, and we only have a minute left, but I want to ask you one question too, and this was brought up a little bit about by Rhonda this morning, but it's really about the consistency of the product that's coming out. I mean, obviously, we're talking about autologous coming in, so there's variability somewhat in the cells, but how often are you able to meet your specs in terms of what you're actually pushing out with product?
Right. Well, it's actually a pretty consistent process, and so, we'll have a few manufacturing failures along the way because we have a pretty stringent product spec that we need to meet, just like you do for other biologics, right? It's around viability, numbers of cells, and so on. And so we have to meet those specs. And obviously, as you move through clinical development, those specs start to tighten. But, we've taken a very good approach. I mentioned that our specs, both in the phase 2 and phase 3 study, were approved by the FDA. But, once in a while, you might find, you might get a fatty aspirate, and so it kind of messes up with the system or messes up your ability to produce as well. But it's really not a very common. And we would expect that at a commercial scale, any kinds of manufacturing issues less than 10%, would be kind of standard course.
Perfect. And with that, we're right on time. Nick, thanks so much for presenting.
Thank you.